Greetings, my name is Paula Gonzalez and welcome to another lecture from the 2020 Honors Chemistry Short Lecture Series. Have you ever wondered what police officers or forensic chemists use to detect drugs that are unknown? Well, today I'm going to be talking about the detection of opioids. This topic of presentation was inspired by the CNN article Power Detection Technology for Powerful New Street Drugs, published in 2017 by Emma Kielski. Throughout this presentation, I will be going over the following: the key points in the CNN article, the effects opioids have on people, the portable instruments that police officers use on the field to determine unknown drugs, the instruments used to test drugs in forensic labs, and finally, the new instruments that are being tested to potentially be used in forensic labs to increase the sensitivity and accuracy of the results. Power detection technology for powerful new street drugs. The article describes various instruments that help detect opioids on the field and in forensic labs. It states that this type of detection is important because opioids are dangerous drugs that were related to more than 63% of the overdose deaths in 2015. The article mentions that new instruments like the TD-DART-MS and the IMS are being created and modified to help forensic chemists and officers stay safe and to be able to achieve the most accurate results. So what kind of effects do opioids have? Well, first let's start by understanding what is an opioid. Opioids are a class of drug naturally found in the opium poppy plant and that work in the brain to produce a variety of effects, which include the relief of pain. Opioids, like one of the most popular, fentanyl, binds to the mu opioid receptor on the cells in the body. When triggering this receptor, it relieves pain and causes a euphoric high. One of the most popular and strong opioids is fentanyl. A dose of 2 to 3 milligrams can be fatal. Although it is a common illegal drug, it is also a Schedule II prescription drug and is typically used to treat patients with severe pain or pain after surgery that is too hard to manage. Fentanyl is also similar to morphine, but is 50 to 100 times more potent and 30 to 50 times more potent as heroin. One of the most dangerous drugs is carfentanil, which is a structural analog of fentanyl, used as a large animal tranquilizer. It started appearing on the streets around 2017, and it is 100 times as potent as fentanyl and 10,000 times as potent as morphine. Some effects that these opioids have are addiction, heart infection, lung infection, heart failure, and muscle pain, as well as feeling of calm, sleepiness, and confusion, slowed or stopped breathing, nausea, vomiting, and constipation. On the right, you see a comparison of 3 opioids. The amount of the drug you see on the picture is the amount that is fatal to the human body. As you can see, carfentanil is the most dangerous, with 2 milligrams being able to harm 100 people. Then fentanyl, and lastly, heroin. On the left, you see 3 methods to consume opioids: pills, injections, and snorting. So what do officers use to detect drugs on the field? The DEA recommends that the first responders who encounter unknown drugs suit up in protective gear to ensure their safety. One of the most common forms of identifying drugs is the wet chem kit. This is a small kit that consists of a plastic envelope containing sealed ampules of various reagents. Inside the envelope, you put in a small sample of the unknown drug and then close the envelope. Then squeeze and pop open the ampules in the designated order and mix the reagents with the sample and watch it change color to find out what type of drug it is. As you can see, the picture on the top is a brand new envelope, and the one on the bottom is one that has been used and is showing that the unidentified drug is fentanyl. Now, this method is really dangerous because it requires a sample of the unknown potentially dangerous drug. There are 2 other popular instruments called the TrueNarc and the Tactic ID, which are a much safer option and a more reliable way to identify the unknown narcotics. The devices collect spectra, produced when a laser light scatters from molecules in the sample, and then they match the spectra to those in a built-in library for various drugs, analogs, drug precursors, and cutting agents. These handheld spectrometers can scan through translucent material, removing the need to open packaging for sample collection, making it a lot safer for the officer. After scanning the unknown drug, the device reads out the highest hazard component. The instrument stores the data from every test until transferred to a computer. There is no way to delete this data, which is a part of the tamper-proof design that meets law enforcement chain of custody needs. The results appear within 30 to 90 seconds after the scan. This is very helpful, especially when you have multiple unknown substances. All tests that are conducted on the field or during an investigation are considered presumptive and can only help the police officer or investigator determine if the drug is illegal or not. The evidence collected is always going to be sent to a forensic lab for confirmatory testing. Once they arrive at forensic labs, the evidence is put through a series of tests and a complete analysis report that can be used in a court proceeding. The analysis in general consists of weight test, presumptive testing or screening, and confirmatory testing. The weight test can determine what the material's net weight is. To weigh the material and record the results, they use an analytical balance. This test also helps determine if the amount of material is sufficient for further testing. Once that is done, they do a presumptive testing or screening, which determines the general characteristics of the sample material. This will help by narrowing down the field of confirmatory tests that will be used. Confirmatory testing is the last test, and this identifies and separates the individual components of the material. This test consists of two parts: the separation of compounds, in which they can use various methods, one being gas chromatography, and the second part is to identify each component, in this case using an instrument like mass spectrometer. The instrument gas chromatography, or GC, dissolves the material in a liquid solvent, injecting the liquid into a superheated oven, vaporizing the liquid and pushing it through a very small and long glass capillary tube using a carrier gas such as hydrogen or helium. The mixture separates into individual chemical components inside the tube. As each component travels through the tube at slightly different speeds, it allows the analyst to measure the time it takes each component to emerge and compare that to reference materials. The mass spectrometry instrument is used after the materials have separated, and it uses beams of electrons that cause them to break apart. Chemicals always break apart in the same way due to their chemical structure, and this can be mapped into a spectrum and compared against materials. In the article, it explains how the U.S. National Institute of Standards and Technology are constantly trying to develop more sensitive methods for testing deadly drugs. They do this by modifying instruments and sampling methods, already in use for detecting trace levels of explosives. For example, the ion mobility spectrometry, or the IMS, and the thermal desorption direct analysis in real-time mass spectrometry, or the TD-DART-MS, are instruments that could rapidly detect trace levels of fentanyl, 16 levels of analogs, and 5 other opioids with a non-destructive wipe sampling. This was revealed in a recent study. Both of these instruments could also reliably detect fentanyl in mixtures with other drugs, even with fingerprint residue and dirt contamination. The TD-DART-MS makes the compound simpler by ionizing the substances and comparing the structures. It uses a helium plasma stream set to a desired temperature and a polarity to vaporize and ionize molecules from a liquid or a solid temperature. It overcomes many issues associated with sample preparation because of its ability to analyze large samples in air and without contamination. It can also analyze a wide variety of liquid and solid samples. The IMS is a technique that differentiates ions based on their size, shape, charge, and mass. A plastic wand which has a polymer wipe at the end of it is used. You rub the head of the wand on a drug sample, and after that, the head of the wand is then placed in a port on the front of the IMS, and the chemicals on the wipe are vaporized, ionized, and separated, with a weak electronic field. The results appear in about 10 seconds. Although these instruments show potential, they aren't quite ready to be detecting opioids in forensic labs. or mobile units just yet. It still needs further testing and validation according to the CNN article. Opioids are a very dangerous and addictive drug that have taken over the streets. Since 1999, opioid-related overdoses have quadrupled. These dangerous drugs can often be a threat to officers when they are trying to determine what exactly is in the unknown drug. This is why the instruments they use are constantly being modified to ensure that they can get the best readings while still being safe. The instruments used by forensic chemists are to officially confirm the components of the unknown drug to be used in court proceedings. However, forensic chemists at times can be overwhelmed with samples, which is why new instruments are being tested, not only to increase the sensitivity reading, but to also be able to get a more detailed and faster result. As for the future, the instruments that are under testing still have a long way to go in determining whether or not they are best suited for detecting drugs. Thank you for listening.